Well, thanks so much for the invitation. So I'm going to talk about COVID-19 vaccine trials, a year of progress. And at Sean's request, I'm going to focus on the perspective of our research unit at the Kaiser Permanente Washington Health Research Institute and the work we've been doing on these vaccine trials. But also we'll discuss other trials and other relevant studies that have produced data during this time period. As disclosures, I've received grant support from the NIH for the conduct of clinical trials for COVID-19 vaccines manufactured by Moderna, as Sean mentioned, Janssen, and HTT Bio. So on March 16th, 2020, this Seattle area resident received the first injection of a COVID-19 vaccine in the world. And that was the investigational mRNA 1273 vaccine co-developed by Moderna and NIH. This vaccine, as we all probably know at this point, encodes synthetic mRNA that codes for the spike protein of the coronavirus, and the spike protein is instrumental in allowing the virus to attach to cells and then to enter cells and multiply. The synthetic sequence is then encased in a lipid nanoparticle capsule in order to allow it to safely travel and to facilitate binding and entering cells. Once in the cell, the mRNA is then translated into a spike protein, which is a trimer, so then that assembles into the full spike protein and is then presented on the cell surface. Importantly, it does not integrate with the cell nucleus at all. There's no risk of abnormalities due to integration of that genetic material. Importantly, this vaccine, as with all the other vaccines that have reached the stage of phase 3 efficacy trials in the U.S., can be manufactured without any access to the actual virus and does not contain any component of the actual virus. All that's needed to start the process is knowledge of the gene sequence for the spike protein for this particular coronavirus. And that was available on January 10th when a Chinese scientist posted the full genome sequence to an online website. From that point, the decision is made on the exact mRNA sequence to encode. That those are synthesized, encased in a lipid nanoparticle capsule, injected, and then travels and enters into myocytes as well as other types of cells. The S protein is created and expressed and secreted, and then activates cytotoxic T-cell mediated immunity, which can directly kill circulating virus, and also stimulates humoral immunity with production of antibodies that can bind to the spike protein and block the virus from being able to bind to and enter cells. This is the first 30 lines of the genomic sequence that was posted on January 10th. And that was used by the Moderna NIH team to start the process of developing this vaccine. The other vaccines that we're seeing entering EUA or nearing completion of phase 3 trials actually all operate by the same general principle. They act to insert the genetic sequence in a genome of a virus or in a standalone mRNA fragment, as with the mRNA 1273 vaccine. And then there's a mechanism whereby that genome gets into a cell and then can be translated into the protein that can then be presented to the immune system. In the case of the adenovirus vectored vaccines like Ad26-CoV-2S, the adenovirus is a DNA virus, so the genetic sequence in this case is a DNA sequence that's inserted into the viral DNA. The adenovirus can bind and get into cells. Once in the cell, the DNA is injected into the cell nucleus. However, there was no integration with the cellular DNA. It's transcribed to mRNA, and then the mRNA goes through the process that we just saw to produce the spike protein. Another vaccine that's probably nearing EUA is a protein nanoparticle vaccine. And this is manufactured in a little bit of a different way in that the cells that produce the spike protein are not human cells. They're not from the cells of a person who's received the vaccine. They're actually insect cells that are maintained in cell culture in the laboratory. The insect cells are infected with a baculovirus, and the DNA of the baculovirus has been modified to include the spike protein insert. So this is the way that genetic material gets into a cell. And then the mRNA is translated, spike protein is produced and expressed on the cell surface. And then that is harvested and purified and made into these lipid particles. That is then combined with an adjuvant, which is an immune stimulating substance. And so in this case, the persons receiving the vaccine do not receive genetic material, but receive the spike protein that was produced as a result of cellular production based on that genetic material. Well, there's a bit of a plot twist here because, I said that the scientists used this sequence that was posted online in order to design these vaccines, and that is true that the genetic sequences used in the vaccines are based on the actual viral sequence encoding for the spike protein, but they are not identical to the viral sequence encoding for the spike protein. And the reason that they're not identical is the result of about a decade or so of work on development of vaccines for other serious coronaviruses, namely SARS-CoV-1 and Middle East Respiratory Syndrome virus, or MERS. And in the course of that work, the scientists discovered, especially in the MERS work, that if you synthetically, or you created a spike protein in the lab and injected that into, say, mice, that it actually was a poor immunogen. And the reason is that this protein adopts different conformations. So a native on the surface of the cell, it assembles as a trimer. The most important part is the S1 component, which is the head that binds to the host cell receptors, the ACE2 receptors. This is the prefusion conformation. And this is what we want antibodies to recognize, because we want to be able to block this binding to the host cell receptor. If the virus does bind, then it adopts these progressive folded conformations. So when the spike protein is created and then just injected sort of on its own into the mouse, it tends to fold down and adopt the same type of conformation as the cell fusion conformation. And you can see that if it becomes folded like this, different epitopes are going to be presented to the immune system. You're not going to get the clean binding of antibodies to the S1, in particular to the receptor binding domain, which is really the part that actually directly encounters the ACE2 receptor. And you may get ineffective antibodies or potentially even interfering antibodies. And through a lot of trial and error work, it was discovered that if you substitute two prolines into the amino acid sequence for the protein, that this is stabilized into this prefusion conformation. And so this work was done by Jason McClellan, who was collaborating with NIH and is now at the University of Texas, Austin, by Barney Graham, who leads the Vaccine Research Center at NIH, by Kismikia Corbett, who's a researcher at that center, Mark Dennison at Vanderbilt, and a whole team working on this issue. So they call this a two-proline substitution spike protein, S2P, and we'll be seeing more of that. And in this experiment in mice, the spike protein was created as the S2P protein and also as the wild type. that was based directly on the actual viral genomic sequence. And the protein then was injected in a small quantity in the mice. And there was low-level neutralizing response to the wild-type protein, but a greater response to the S2P protein in these mice. And then with increasing dosage, you get some increase to the S2P, but you see kind of a linear increase to the wild-type. And it's only when you go up 100-fold to the dose that you get an equivalence between the neutralizing activity to the S2P as opposed to the wild-type. This work was published in 2017, and the authors offered a very prescient conclusion to this manuscript, where they said, collectively, these results advance our understanding of MERS-CoV entry, and antibody-mediated neutralization, and provide a foundation for the structure-based design of vaccine antigens for highly pathogenic coronaviruses, including those expected to emerge in the future. And so that's exactly what happened. When the NIH and Moderna teams were working to develop the mRNA sequence to include in the vaccine, they relied on this work, and they included the S2P substitution as the protein that the mRNA would encode for. As part of the preclinical work, additional experiments were conducted, also using the MERS model. But this time, instead of injecting the mice with the spike protein, they injected the mice with the mRNA encoding either the S2P protein or the wild-type spike protein. And here we see, if you use small amounts of mRNA, you get no neutralizing response to the wild-type, but a pretty vigorous neutralizing response to the spike protein that's statistically higher than that to the wild-type. And the same kind of pattern where you need to go, fold increases higher for the two to be equivalent. And here, the neutralizing activity to MERS is relatively insensitive to increasing dosages. So you can obviously use a lot less mRNA to produce the same level of neutralizing activity. So that has formed the basis for preference toward the S2P, and that is incorporated into all of the vaccines that utilize this sort of genomic transfer mechanism and that have made it to the phase 3 trials and beyond that we're seeing now. So you might wonder how did it happen that our institute at the Kaiser Permanente Washington Health Research Institute managed to be involved in giving the first ever SARS-CoV-2 vaccination to clinical trial subjects? And the answer is that the reason that we were able to do that is that we're part of the NIH-funded Vaccine and Treatment Evaluation Units, the VTEUs. This is a network that's been continuously funded by the NIH since the 1960s. Our mission is to evaluate diagnostics, therapeutics, and vaccines for infectious diseases of public health importance, and to maintain pandemic preparedness against emerging infectious diseases. And so we were called into battle for the 2009 H1N1 pandemic. And especially because this vaccine was co-developed at NIH, the decision was made that the first trial would be conducted in the VTEU network. And we were fortunate enough to be the site that was selected for that trial. I should also add that of the 10 sites in the U.S., 2 are in Seattle, so go Seattle. As far as the timeline, we see here that January 10th was the day that everyone got a first look at the SARS-CoV-2 genome. 3 days later, NIH and Moderna had finalized the mRNA sequence for the vaccine. We were selected at the end of January, and it's been pretty much a full-on sprint ever since that time. The first vaccine batch was manufactured extremely quickly by February 7th, went to NIH as a study product available for the trial. The necessary FDA review was completed on March 4th, which meant that we could proceed ahead with the trial. So we started recruitment and screening of volunteers and gave that first injection on March 16th. So this is 45 days since the posting of the genome to the availability of vaccine study product, and 66 days until the first volunteer was inoculated, which of course is just, orders of magnitude more rapid than any normal study conducted in the before times. This study design was initially designed as a single-site trial to enroll 45 healthy younger adults, 18 to 55, who would receive one of 3 dose levels of the vaccine, 25 micrograms, 100 micrograms, or 250. And this went through a dose escalation design whereby the first the first group of participants received the 25 microgram and then went up from there. Everybody received 2 vaccinations given 28 days apart, as is familiar now from the EUA schedule. But as we've continued to see as we've gone through these processes, there were changes that happened, changes and adaptations. During the trial, the decision was made to enroll older adult participant cohorts, persons over 55, and 2 additional sites, Emory University School of Medicine and the NIH Vaccine Research Center, were added in order to increase capacity for enrollment and also to give some, situational capacity in the event that an outbreak at any given location would reach enough severity that it could impair our ability to continue to conduct the trial. And this was highlighted because, as you all might remember, Seattle was the first real hot zone for SARS-CoV-2, and that pretty much coincided with when we started this trial. In July, we published the first interim report from the 45 younger adults who had received both vaccinations. The safety data indicated that this vaccine is reactogenic, especially with higher doses and after the second vaccination. So if we're looking at any systemic symptoms here, we can see that the majority of people, especially with the 2 higher dose groups, had some sort of systemic symptom. Most were mild, as indicated by the brown. Fever did not occur. Local symptoms were common, especially pain, and in the higher dose groups, most persons had some degree of pain at the injection site, but it was typically mild. After the second vaccination, systemic symptoms were much more common, especially in these 2 higher dose groups, when 100% of participants had at least one systemic symptom occurring in the post-vaccination period. Fatigue was very common. Here we now see fever in these 2 higher dose groups with 1 episode of severe fever, with temperature over 39 degrees C. Chills were very common, occurring in 80% or so of participants. Also headache with the highest dose, 100% of people reported headache. Local symptoms were again common, with pain being noted by everyone in the 2 higher dose groups, but typically mild or moderate. Looking at immune responses, this is ELISA measured binding antibody, so just quantification of antibody that binds to the S2P protein. We see some increase after the first vaccination in these groups at day 15, but really it's after the second vaccination when you see a booster response and much higher levels of antibody titers. There's also a dose effect with higher doses in the 100 compared to the 25 and the 250 compared to 100. Here we compare with the values in a panel of convalescent sera from persons that have recovered from COVID. And we can see that these binding antibody titers were quite high relative to the convalescent sera and were in the upper quartile of those values, indicating that the immune responses to the vaccine were at least equivalent to those as induced by the natural infection. Here we're looking at neutralizing activity, and this is a functional activity, the ability of the vaccine-induced activity to prevent the virus from adhering to and entering us and infecting cells. Here we don't see much after the first vaccination. It's really not until after the second vaccination when you start getting a personal neutralizing activity. And there is this dose effect with a 2.5-fold jump from 25 to 100, and then a smaller degree of increase from 100 to 250. And now here in the upper half of the values reported in the convalescent serum. So this was, very positive. I mean, this was a new vaccine to a new virus, and we weren't sure, how well it would do. So this was very good news to see these kinds of responses, especially in comparison with the values in convalescent serum. So now I'm going to talk about the elephant in the room and the question you're probably all asking yourself. And I can say, yes, it's true. This work was funded in part by Dolly Parton. And of course, that's now quite well known. She made $1 million donation to Vanderbilt, and that funded some of the neutralizing assays that were employed during this study. These interim findings did support advancement of vaccine to later stage trials. Unlike at traditional patterns of trials, when you finish one trial and then move to another and so forth, that's not the way this occurred. These interim findings were taken and run with in order to plan and institute a phase 2 trial, which evaluated doses of 50 micrograms and 100 micrograms, and that was initiated in May. And the phase 3 trial of 30,000 adults enrolling at 92 sites in the U.S. Was to evaluate the 100 microgram dose, which of course had to be selected very rapidly in order to manufacture enough of that vaccine to provide to the 15,000 people who would be randomized to vaccine. And that was initiated on July 27th, 2020. The 100 microgram dose was selected for the phase 3 because the 250 microgram dose seemed to have more reactogenicity and didn't provide really appreciable increase in immunogenicity. So it seemed like the optimal of the 3 doses to pick for to go to phase 3. The 50 microgram dose was selected for the phase 2 because the 25 seemed too low and the 100, seemed best of the 3, but maybe, an in-between dose of 50 micrograms could provide comparable immunogenicity and possibly, somewhat less reactogenicity, but also be dose sparing in terms of the amount of vaccine material that you need to have would be cut in half if you could use a dose of 50 instead of a dose of 100 micrograms. We enrolled the older adult cohorts, and here in September report interim findings from that group. There were divided into cohorts of 56 to 70 years of age and 71 years of age and older, and received either 25 micrograms or 100 microgram 2-dose series. And looking at the neutralizing activity, we can see that here again, really nothing after the first dose, but you do get some activity coming on after the second dose. The activity titers were lower with the 25 microgram than with the 100 microgram, supporting the use of 100 micrograms. A really interesting finding here, though, was that when you look, say, across the 100 micrograms, there's no age effect. The titers induced are really pretty much identical in the younger group and in the older group. And then if we bring in our previous data from the responses to 100 micrograms in the younger adults at day 43, you also see that there's no decrease across with increase in age from that group. So from adults 18 and older, the results are constant across age, which is pretty remarkable and unexpected, I would say, because our past experience with, say, influenza vaccine is that you start seeing declines in immune responses, and starting with adults in their 30s or 40s. And then with each subsequent decade of life, you see further and further declines. So the reason for this, very good finding is unclear, but possibly because persons are basically naive to this virus, and maybe there's an opportunity for them to induce a more pure, immune response on first presentation of the spike protein. Or maybe it's because of the structure developed design that, ensured that we would be presenting a conformationally correct spike protein to the immune system, and so that there would be less of a chance of dysfunctional or aberrant immune responses. So we don't know, but anyway, this is a good thing. So going back to our timeline here, the phase 1 was complete for the younger adults enrollment in April. In May, the phase 2 started. In July, the enrollment was completed in the phase 2. And then at the end of July, the phase 3 study began. So now we're looking at a period of only 4 months between giving the first injection in the phase 1 and starting the phase 3, a process which normally evolves over the course of many years or even decades. The first interim efficacy analysis results were announced on November 16th, indicating that the vaccine had an incredible 94% efficacy, which was essentially identical to that reported a bit earlier by Pfizer for their mRNA vaccine. And as with the other vaccines, another strong finding was that efficacy against severe disease requiring hospitalization or leading to death was 100%. There were no such cases in the vaccine group as compared to 30 in the placebo group. So really just an amazing success story for the phase 3 study for both of those vaccines. And the EUA was then granted in December 18th, 2020. Vaccine was rolled out and injected into arms before the end of the year. So since that time, we've seen hundreds of thousands of doses being administered, achieving coverage levels of upwards of 20% overall in the population. And we've seen additional research come out and produce some recent relevant findings. So I'm going to talk about some of those studies and results. Some reports of real-world vaccine effectiveness of mRNA vaccines, the durability of neutralizing antibody activity, the interim results that have now been reported for that phase 2 study, and then what happens when you give vaccine to someone who's already had COVID infection. This is a study from CDC that involved utilizing a prospective cohort of healthcare workers that had been set up prior to the availability of vaccine and had been followed weekly with PCR testing to identify symptomatic and asymptomatic SARS-CoV-2 infection. They were then followed through the period in which vaccine was introduced and for another couple of months. And person time during that period was classified as days that people were unvaccinated, days that they were partially immunized, having received the first dose 14 or more days before, but not having received the second dose, and fully immunized, being they were now 14 days after the second dose. And so as this cohort was followed at the 8 U.S. locations, these analyses determined that full immunization resulted in a vaccine effectiveness of 90%, so pretty much mirroring what was seen in the phase 3. And partial immunization resulted in vaccine effectiveness of 80%. So there is some protection induced by the first vaccination, with the caveat that we don't know how long this would last, how durable it would be if people never end up getting the second vaccination. But anyway, some immunity is induced in the interval before they can reach the state of being fully vaccinated. For antibody persistence, this is, again, data from the phase 1 study. This is looking at activity going out to day 209, which is 180 days after the second dose, so 6 months after the second dose of vaccine. And this is looking at the 3 different age cohorts, 18 to 55, 56 to 70, and 71 plus, of persons who received a 100 microgram dose. And what we say, looking at receptor binding domain ELISA, looking at antibodies that bind to the receptor binding domain of the spike protein, there's not very much degradation over this time, and there's no difference between the age groups. So this indicates, consistent, durable binding antibody responses anyway. With a neutralizing assay, the slope's a bit greater, and we do see some diminishment of neutralizing activity. However, there is still neutralizing activity persistent at day 209, and that those levels did not vary depending on age. So as was seen with the earlier results, comparing younger and older adults, here with these later time points, we still see that there's no difference by age. So these findings do kind of match up with what Moderna just recently reported in a press release, that when they were looking at the data that's now available as the phase 3 participants have progressed in time following vaccination, where people have now been a median of 6 months since the second vaccination, that they're finding 90% effectiveness at protecting against symptomatic COVID-19 and 95% effectiveness at protecting against severe disease. So that's now based on a total of 900 cases and 100 severe cases, and indicates that there's not been an apparent loss of effectiveness over this time period. However, we don't know what will happen as more time goes on. And so there's a lot of talk of booster doses being administered perhaps on an annual basis or something like that, either because there's declining immunity to the original vaccination or because there's diminished effectiveness against variant strains. And so either or both of those rationales might call for the use of subsequent booster doses. So in response to this, we've modified the phase 1 study to now include an optional third mRNA 1273 vaccination for those participants who reach, the 9 months or later time point after their second study vaccination. And so people that have received both of the planned initial vaccinations can choose to enter the sub-study and receive a third vaccination and then be followed for 1 year after for safety and immunogenicity. And we've enrolled about 60 of our participants in this sub-study, and Emory University, which was one of the other sites, is also enrolling participants into this sub-study. As far as the phase 2, these interim results have been recently published. Again, this was looking at 2-dose schedules of 50 micrograms or 100 micrograms in adults 18 and over. And here we see that in the younger age group of 18 to 54, there's really no difference between the neutralizing responses to 50 micrograms or 100 micrograms, looking at time points after the second vaccination. Similarly, in 55 plus, no real difference between 50 and 100 micrograms, and in both cases higher than the values in the convalescent sera panel. And again here, what we also see is there's no difference by age. It's pretty flat across both dose groups and both age groups. Lastly, looking at people who were seropositive, indicating that they had been previously infected prior to vaccination, and comparing their vaccine responses with persons who were seronegative. The seropositive are in the yellow here. And we see that persons who were seropositive before the first dose tended to mount a pretty quick antibody binding response after the second dose. Within a week, they had pretty high binding antibody titers as compared with a lack of response at this time point among the seronegative group. Then the seronegative group started to pick up at later time points, whereas the seropositive group, quickly reached this level and then really didn't progress much beyond that at later time points. Right here is the last time point before the second vaccine was administered. And you see some increase after the second vaccination with the naive group, but really nothing with the seropositive group, suggesting that essentially the seropositive people had received their first vaccination as a result of their COVID exposure and are reacting here as a booster, receiving their second exposure in the form of vaccine, but receiving a third exposure within this interval isn't going to bump them up any higher, whereas the naive people were reacting to the first dose and then received some boost to the second dose. A common question has been, what about the side effects? If you've had COVID and you get a vaccine, are you more likely to have side effects than a naive person? And the answer is that does seem to be the case. Not so much for these injection site reactions. Maybe there's a bit more pain, but that seems to be sort of irregardless of whether you've had COVID before. But certainly for the systemic reactions, we can see here that clearly the systemic reactions were more common for fatigue and headache and chills, muscle pain, fever, in persons who'd been previously infected compared to persons who hadn't, which does mirror the experience of these signs and symptoms being more common with the second vaccination dose in seronegative persons as compared with the first vaccination dose. So here we are where we're getting all this information, we're rolling out vaccines, people are getting protected or saving lives, and everything's rolling along, except that now we have to deal with variants, which are rapidly emerging. Here are the CDC definitions for variants. A variant of concern is one at which you know or you have evidence that there is more highly transmissible, causes more severe disease, leads to reduction in the ability of antibodies induced by vaccine or previous infection to neutralize the variant virus compared with the original virus, and reduced effectiveness of treatments or vaccines or diagnostic detection failures. And variants of interest are ones that you suspect they might have some of these components because they have some of the same genetic markers that are associated with variants of concern, but they've not yet been demonstrated to have reduced neutralization or reduced efficacy of treatments or vaccinations or increases in transmissibility or, disease severity. Our situation in Washington State currently is that variants are widely circulating. Here we see back in February, this other category means they are not variants of concern or interest, and they are largely strains and lineages that are relatively closely related to the original strains that were circulating early in the pandemic. And that they, at that time, accounted for the majority of strains. However, there's been a clear progression over time, and these are the last, most recent data available, in which those sort of earlier lineages are now only about 15%, and 85% are made up by variants, all of which are variants of concern, including the B1351, which initially emerged in South Africa, P1, which initially was identified in Brazil and Japan. These two, B127 and 1427 and 1429, emerged out of California. And then the B117 is becoming more prominent here, and it originally emerged out of the UK. Of these, I mean, they're all of concern, but the one that is arguably of highest concern is the B1351 variant. This variant has 8 mutations in the spike protein, including the so-called EAT mutation, which is associated with more severe disease and higher transmissibility. This variant is associated with about a 50% increased transmission compared to earlier lineages. It is associated with reduction in neutralization by some monoclonal antibody treatments. And there's been identified moderate reduction in neutralization by both convalescent sera and post-vaccination sera. So all these factors are causes for concern for this variant becoming more widespread. And it can take over pretty quickly. In South Africa, it was first identified in October. By the end of November, it accounted for 95% of all strains. As far as nomenclature, in the South African literature, you often see this S501YV2, which is the next strain clade nomenclature. In the US, we tend to use this pangolin lineage nomenclature and say it's B1351, but they refer to the same thing. This is put out by CDC. The B1351 variant has spread worldwide and is being identified in more countries as the weeks go on. This is data from the phase 1 subjects from sera samples collected from participants who received their second 100 microgram mRNA 1273 vaccination, and comparing the ability of that sera to neutralize SARS-CoV-2 virus of an earlier, early pandemic strain or one of the variants. And here we look at the early D614G strain, which emerged pretty early on as to be the predominant strain in the US. And there was high neutralizing activity against that strain induced by vaccination. And to the B117 variant, which is associated with increased transmissibility, but hasn't really been shown to have evaded vaccine-induced responses, as is confirmed here, with the neutralizing activity essentially not changed, no significant difference between the activity to these two strains. But in contrast, we looked at here at B1351, you do see with each individual person's sera sample, that sample's activity against the original strain is much higher than that same sample's activity against the variant. And in fact, there's a mean of about a 6.4-fold reduction in neutralizing activity to the variant compared to the original strain, indicating a real concern about how well these vaccines are going to protect against particularly this variant strain if it emerges more widely in the US. And there is evidence that this strain is associated potentially with lower vaccine efficacy. These data are from phase 3 trials of the protein nanoparticle vaccine that were conducted in both the UK and in South Africa. The time point in which persons had been enrolled and were being followed for infection was during the time in which this variant had taken over in South Africa. And so nearly all of the strains identified from vaccine trial participants were this variant strain. And the investigators in an interim analysis found that for HIV negative persons, some HIV positive people were included in the trial who were baseline seronegative, had not apparently been infected prior to being enrolled in the trial. The vaccine efficacy was only 60%. This is compared with a vaccine efficacy of close to 90% in the UK trial during roughly the same time period, during which time both the original strain and the variant strain were circulating. And there's, a suggestion that maybe there's a somewhat lower efficacy against the variant strain, but probably not drastically. So it's essentially maintained to the variant strain as compared with the original strain. But these estimates of, 96% or 86% are obviously quite a bit higher than the estimate of 60%. suggesting that existing vaccines may not operate as well against the variants should they occur. So this has prompted us to now be conducting a new trial of a variant vaccine manufactured by Moderna, in which the mRNA in the vaccine encodes for the spike protein of the B1351 strain instead of the original strains, and still incorporates that 2-proline substitution that we discussed earlier. This will include 2 cohorts, one of which are persons who are naive, who have not been infected or received COVID-19 vaccine, and one includes previously vaccinated adults. And those previously vaccinated adults are the phase 1 participants, once again. So you're probably wondering, wait, you just said that there was an optional third dose booster sub-study for these phase 1 participants, and that is true. Participants who are eligible for both that booster sub-study and for this new trial could decide which one they would like to participate, or do not have to participate in either one if they did not want to. But we have enrolled participants in this new study who then receive a single dose of 50 micrograms of the variant vaccine, or a combination of 25 micrograms of the variant and 25 micrograms of the original vaccine. The naive group includes 150 participants who will be assigned to one of these 8 arms, which are evaluating a variety of different strategies and scenarios, one of which is that people get the EUA vaccine followed by a variant boost. And the EUA vaccine can be what is now being administered, or we're anticipating the possibility at some point that there may be a switch to a 50 microgram dose. We're evaluating both those doses throughout these cohorts in the naive group. And then other people are going to get a primary series of 2 vaccinations, either both the variant at dose of 100 or 50, a combination of original and followed by variant at 10 and 50, and then these combination vaccines that include either a total of 100 micrograms or a total of 50 micrograms, including both the variant and the original vaccine. Well, we've now covered from the beginning of all of this to the present day, and it certainly has been an extraordinary period in which tremendous progress has been made, and there have been many successes. And I especially want to call out our entire Seattle biomedical community that I think has really just stepped up and performed remarkably across the board, including in areas of vaccine development and test and evaluation, in development and evaluation of various novel therapeutics, in development and implementation of diagnostic testing, which has been quite an amazing story, in epidemiologic studies, in disease modeling work that's been done to provide information for use by our leaders locally and nationally to use in guiding their decisions regarding pandemic strategies, in clinical care, in viral sequencing and molecular epidemiology, in communications about our findings and their significance, both locally and nationally. I think it's been really a banner year for Seattle research. But we still have work to do. We're facing new challenges that are going to probably need new solutions. And so we're not done yet, and this story continues. Thanks so much for your attention. And I think we have time to answer some questions if any have come up in the chat.
Great, Lisa. That was really tremendous. It's certainly been a crazy and busy year. There's a few questions in the chat, and I can read them off. And then if other people have questions, they're welcome to add them to the chat as we go along. We have a few minutes here. So the first question was from Mark Winter, and he said he's interested in immunosuppressed people. So your thoughts and recommendations on immunizing immunosuppressed people. How do you monitor their response, and what do you do if they don't respond?
Well, this is, a common and understandable question that comes up. Unfortunately, we at this time don't have a lot of answers. We see in the South African trial that the limited number of persons living with HIV that were enrolled did seem to not have as much protection as non-HIV infected persons. But the numbers are small, and that's, there are many groups of immunocompromised people, of which that is one that maybe is not cannot be extrapolated from to other groups. We don't know. We don't really have experience with this type of vaccine, the mRNA vaccines, in immunocompromised people. It's possible that they may outperform, more conventional vaccines, such as flu vaccine, for example, which does appear to have decreased effectiveness in immunocompromised persons. What to follow is also difficult. In the context of a trial in which we can compare, contemporarily collected samples from before and after vaccination and from non-immunocompromised and immunocompromised groups, we could draw some conclusions. But just for an individual person to receive a vaccine, at this point, we don't really think there's a lot of value in attempting to go out and have antibody testing done, because it's very unclear at the individual level what that would mean. And then there's a lot of differences in the various assays that are being used, and their accuracy and what they're actually measuring. So I think that's something that we're going to need to make some progress on as we move forward.
All right, there's a second, there's a few more questions in here. One quick question was, did you have the experience at all where people in the placebo control groups of your trials dropped out when there was an EUA approved so they could go get vaccinated, or did you have some way to retain them and work around that?
Oh, we definitely had that. And we did initiate a program of unblinding people. So, at a certain point in time, after the interim analyses were performed, of course. And so people could remain in the study, but those who were determined to have received placebo could then receive vaccine administered through the study and not having to go out and get EUA vaccine. But in advance of having that set up, people, especially people who are healthcare workers or, at high risk of exposure, could sort of self-identify as probably not having received the vaccine if they had no reactions. Now, there is quite a bit of individual variability, and you can have either none or just very, very mild adverse events even after the second vaccination, or you can, have more severe events. So it's not a perfect self-identification measure, but we did have that happen, and it did end up that some people who had received the vaccine actually ended up getting EUA vaccine as well, which is not optimal. But by and large, it was controlled by this setup system of being able to unblind people within the study.
A couple of people have asked whether T-cell responses were monitored, and do you have anything to say about that?
Yes, let's see. I actually have a slide on that. I'm going to need to. It's hidden inside. Well, I'll just say that I did have a slide, which I can't get unhidden right now. But yes, we did look at T-cell responses, and the results were encouraging. There were strong Th1 responses and really no Th2 responses. So another concern has been, whether these vaccines could possibly induce enhanced disease following vaccination, and the finding of strong Th1 and not Th2 responses provides some evidence that that's probably not going to be the case, as well as our ongoing follow-up of people in these trials and in the phase 3 trials has not produced any evidence of an enhanced disease as well.
I have a question to add, which is an advice question. We can spend a lot of time talking about the differences between the vaccines and ways that we can make them better, and that's like the scientist part of us. And then at the same time, the public messaging is to please go get vaccinated with what's already available. It's sort of like two hats. Do you have any advice to healthcare providers about how to both be investigative and to urge vaccine uptake at the same time?
Well, I think we're in a fortunate position in that all the vaccines seem to be very strongly protective against severe disease. So, and that's really the most consequential outcome, right? And that's what we want to prevent is people winding up in the hospital or dying as a result of SARS-CoV-2 infection. And, of course, as we accrue more and more information, there will be some cases that occur in the vaccine group. But in the interim analyses, I think it was pretty consistently found that there were no deaths due to SARS-CoV-2 among persons in the vaccine group as compared to the placebo group. So that can argue that, really you should get the vaccine that you're able to get. Now that things have really loosened up, there's a lot larger vaccine supply, and, we've got a lot greater vaccination capability. Maybe people can make their own choices, which is fine, I think. But we want to just get the greatest number of people vaccinated that we can.
I think we'll take one more quick question and then we'll wrap up for the sake of time. Chihiro Morishima asks, based on the available data, do you think that people who have had COVID need 2 doses of the vaccine?
No, I would think that, well, with the caveat that, maybe it depends on the severity of disease or how long it's been. If it's been a long time, then you're really looking at maybe a booster response, but probably, within the time frame of the pandemic and the availability of vaccines, it's probably fine for people to get one. But as long as you're willing to risk some adverse events, it's not that much of a downside to getting a second one just to be sure. But this is something that we'll probably be getting a lot more information about to allow us to make, a firmer statement about the use of vaccine in persons who've been previously infected.
Great. Well, Lisa, on behalf of our whole department, I just want to thank you for all the work you've done and for sharing it with us today. We really appreciate it.
It's my pleasure. Thanks so much for inviting me, Sean.
Great. We'll see you later. Thank you, everybody. Have a good day.